So today I'm going to be talking with you about cellular respiration, and this is the process cells undergo to produce energy in the form of ATP. And there are two pathways a cell can take: anaerobic, which is this process without oxygen, and aerobic, which is this process with oxygen, and that's what I'm going to be showing you right now. So there are basically four major steps: glycolysis, pyruvate oxidation, citric acid cycle, oxidative phosphorylation, which includes electron transport chain, and chemiosmosis. And three of these cycles produce ATP, which will result in about 30 or 32 ATPs, and all but glycolysis occur inside the mitochondria. So the first step is glycolysis, which is the decomposition of glucose to pyruvic acid, or pyruvate. In actuality, there are 10 steps to this process, but you really only need to know the information in this video for the AP exam. Glycolysis can be subdivided into two stages, and the first one is the energy investment phase, in which two ATPs are used and converted into ADP and phosphate. So what happens is a 6-carbon glucose molecule is converted into two 3-carbon molecules by the destruction of ATP. So the second stage of glycolysis is the energy payoff phase, and this is when NAD+ is transformed into NADH, and 4 ADP plus 4 phosphates are turned into 4 ATP. So in this stage, the 3-carbon molecule is transformed into a pyruvic acid. The next stage is pyruvate oxidation, and this is when pyruvate is modified by coenzyme A and turned into acetyl coenzyme so that it can kickstart the Krebs cycle, or citric acid cycle. This process results in one NADH molecule and one CO2 being released, but it does not result in ATP being produced. So glycolysis takes place outside of the mitochondrial membrane in the cytosol. So in order for the pyruvate to get inside the mitochondria, it has to have the help of transport proteins. The C-double-O group at the top of the pyruvate has little chemical energy, so it is removed and released off as CO2, and NAD+ is reduced and turned into NADH plus hydrogen. So the remaining 2-carbon fragment is oxidized, forming acetate, and finally the coenzyme A attaches via its sulfur atom to the acetate, forming acetyl coenzyme A. So the next stage is the citric acid cycle, also known as the Krebs cycle. And the Krebs cycle it functions as a metabolic furnace that oxidizes organic fuel derived from pyruvate. And just like glycolysis, there are many steps in this process, 8 of them. However, you only need to focus on really the beginning and mostly its products. So what happens is, in the beginning of the citric acid cycle, it begins when acetyl coenzyme A combines with oxaloacetate to form citrate. And then there are 7 intermediate products, and one cycle produces 3 NADH and 1 FADH2 and 1 ATP, and 2 CO2 are released. So the final step in aerobic cellular respiration is oxidative phosphorylation, and this is composed of electron transport chain and chemiosmosis. So like the Krebs cycle, the electron transport chain takes place inside the mitochondria. So this is the inner membrane of the mitochondrion, and it is a double phospholipid bilayer. And so the electron transport chain is a sequence of 4 electron carrier molecules, or membrane proteins, that shuttle electrons during the redox reactions that release energy, used to help establish an electrochemical gradient across the inner mitochondrial membrane. NADH and FADH act as electron transporters. In complex 1, electrons are passed from NADH to the electron transport chain. NADH is oxidized to NAD, and FADH is oxidized to FAD in complex 2. At complex 3, no electrons are added, but electrons still flow through the electron transport chain. In complex 4, the final electron acceptor of the electron transport chain is oxygen. The 1/2 O2 accepts the 2 electrons, and together with 2 H+ forms H2O. The movement of electron causes protons, hydrogen atoms, to be pumped out of the matrix into the inner membrane space. So next is chemiosmosis, and the energy from the electron transport chain enables H+ ions or protons to be transported from the matrix to the inner membrane space by protein complexes. As the protons are transferred, it creates an electrochemical gradient across the membrane. The concentration of H+ further decreases as H+ combines with oxygen to form water. The H+ ions flow back into the matrix via ATP synthase, a channel protein in the inner membrane, and the electrical gradient produces the energy for ATP synthase to generate ATP from ADP and phosphate. So, in general, glycolysis will produce 2 ATP, the citric acid cycle will produce another 2 ATP, and the oxidative phosphorylation will produce about 26 or 28 ATP, resulting in 30 or 32 ATP. So next we'll be talking about fermentation, which is basically cellular respiration only without oxygen, and there are 2 types, and the main goal is to process and replenish NAD+ so that glycolysis can proceed once again. And the first form is alcohol fermentation. So the first step in alcohol fermentation is to turn the pyruvates formed from glycolysis into acetaldehyde, and for each pyruvate, 1 carbon dioxide molecule and 1 acetaldehyde are produced. Next step is to turn acetaldehyde to ethanol, and the important part of this step is that the energy in NADH is used to drive this reaction, releasing NAD+. For each acetaldehyde, 1 ethanol is made and 1 NAD+ is produced. The task of freeing NAD+ allows glycolysis to continue. The second form of fermentation is lactic acid fermentation, and there is only 1 step. After glycolysis, the pyruvates formed are converted to lactic acid, and in the process, NADH gives up its electrons to form NAD+. As an alcoholic fermentation, the NAD+ can now be used for glycolysis. So thanks for watching. We hope you learned a lot and that you'll do well on your AP test in May.